Hello, 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 good afternoon. Sorry, good evening. Hello, hello, hello. Can you hear me? Am I audible? Am I audible? Hello, am I audible? Okay. So, welcome to Goscore Learning. We are the fastest growing edtech in GCC, and again, I am here with the live session of the same chapter that we were discussing previously, that is molecular basis of inheritance. And this is the third session, and we'll try to conclude today of this chapter. And today we are going to discuss the basic concepts of transcription, translation, and about the gene regulation. In gene regulation, we have to discuss about lac operon. Okay, so we'll be discussing with, first we'll discuss with transcription. Dear students, in the previous class, we have discussed what is the structure of DNA, what is DNA, what is RNA, and how can we identify DNA and RNA. What are the major differences between this DNA and RNA? DNA means it's simply deoxyribonucleic acid, and RNA means it is simply ribonucleic acid. The major difference is between the second carbon position, the oxygen group is not present in the case of DNA, whereas in the RNA, the oxygen group is present, OH group is present at the second carbon. Okay, that is the major difference, and from this difference, you understand that there are some chemical changes that may happen in DNA and in the RNA. And also you have learned in the case of DNA, there are different nitrogen bases. Okay, nitrogen bases present in DNA and RNA is different. If you are considering DNA has adenine, thymine, guanine and cytosine. Whereas in the case of uracil, there is no thymine. Instead of thymine, what is present? Uracil is present. Okay, so that is the basic difference between the DNA and RNA. Moving to DNA and RNA, we have learned how this DNA is wrapped inside the nucleus, or how the DNA is present inside the nucleus. Okay, DNA can be wrapped into a smaller structure known as a chromosome. How it is wrapped? It is wrapped with the help of different proteins, and that protein molecule is known as histone protein. What is that protein molecule? It is histone protein. We have discussed the structure of different what we can say nanometer structures of this DNA and how it becomes into a chromosome. Okay. After that, we have discussed what different experiments over how this DNA, how it is proved that DNA is the genetic material. Okay. So today, and also we have discussed about replication. Replication simply means from DNA, what is formed? DNA itself will be forming with the help of different enzymes. Okay, the same process with some little differences is going to happen in the case of transcription. What is transcription? So let's move on. Transcription. Okay. So let's study about transcription. Here I have given the definition for transcription. See, what is transcription, dear students? Transcription is the process of copying. It is the process of copying genetic information from the strands of DNA into RNA, which simply means, that means if there is a DNA present, okay, if there is a DNA present, from this DNA, what is synthesized? RNA is synthesized. Okay, what is synthesized? RNA is synthesized. Specifically, we can say mRNA is synthesized. Okay, what is synthesized? mRNA will be synthesized. Okay. So, you understand what is the meaning of transcription? What is transcription? You have a double-stranded DNA here. After replication, you got the double-stranded DNA. We can mark the position to that is, this is 3' 5' direction. Okay, this is a strand with 3' 5' direction, whereas this is the strand with 5' 3' direction. Okay, this is a strand with 3', 5', whereas the next strand will be 5' and 3'. Okay, so what is the speciality here? I told you in the case of DNA, always think that in the case of DNA, DNA has the different nitrogen bases. Okay, DNA, if you are considering DNA, DNA has different nitrogen bases as adenine, okay, adenine, then thymine, guanine, guanine, and cytosine. Okay, these are the nitrogen bases which is present in the case of DNA. But if you are considering RNA, after transcription we will get RNA. Okay, so this RNA will contain adenine. It will contain adenine. Okay, instead of thymine, what is present? Uracil is present. Okay, what is present? Uracil is present, and this is very important. You have to understand the difference between DNA and RNA. Uracil is present. Okay, then what is next? We can say guanine is there. Okay, then cytosine is there. Okay, cytosine is present. So these are the nitrogen bases which is present inside the RNA. So the next point given here, is, see, adenine pairs with uracil. Okay, adenine pairs with uracil instead of thymine. We can say adenine can pair with thymine. Usually adenine pairs with thymine. In the case of DNA, we can say adenine pairs with, okay, adenine pairs with thymine and guanine pairs with cytosine. So, in the case of RNA, usually adenine pairs with uracil and guanine pairs with cytosine, and cytosine can pair with guanine too. Uracil can pair with adenine too. Okay, this basic concept you have to keep in mind. With this, we will be moving on. Okay, so what happens in transcription? We have to study what happens in transcription. So let's see. Okay, so both strands are not copied during transcription. So in the case of replication, you already know replication, both the strands will undergo replication to form a new strand of DNA. In the case of transcription, these two strands won't be undergoing the process of transcription. Only one strand will be undergoing transcription. Only one strand. Why only one strand is undergoing this transcription? It is because if two strands is undergoing transcription, it will form two strands of RNA that may cause the complementarity. Okay, so if two strands is synthesized, that may cause complementarity. If complementarity is there, it will form a double-stranded RNA. See, if the strand undergo transcription and forms two strands of RNA. See, after transcription, what is the process? That it is translation, right? The formation of protein is known as translation. So, the process of translation won't happen if the strand is double-stranded. Okay, if the strand is double-stranded, this translation process, the process of formation of protein won't be happening. Okay, then the code for protein is different in both strands. See, if you are considering the two strand, one strand is having one particular sequences, then the next strand will have the next complementary sequences. So, if the protein is forming, or if you are considering a particular protein, say for example, we can say an enzyme. Okay, salivary amylase is an enzyme, right? So this salivary amylase can be synthesized only from one particular strand. It cannot be synthesized from two different strands. So if two different strands is undergoing that means transcription, what happens? That protein won't be formed. Okay, so those 2 points, it is very important. You have to keep in mind. That is, the code for protein is different in both the strand. This complicates the translation. Okay, second point. If two molecules are produced, if two RNA molecules are produced simultaneously, it would be complementary to each other. It may be complementary to each other. And what happens here? Hence, it forms a double-stranded RNA. This prevents translation. These 2 points are very important. You have to take a note on this. Okay, question may ask from this particular session. Okay, then next one. What is the transcription unit? Okay, we have to learn about a transcription unit. Dear students, you know that DNA, the structure of DNA, right? DNA is double-stranded. Okay, that also you know. So let's draw the structure of DNA here. Okay, so we have one strand of DNA here. We have another strand of DNA here. Okay, so let's say this is 3 prime, 5 prime strand. Okay, and this one will be, obviously it will be 5 prime, 3 prime strand. Okay, obviously it will be 5 prime, 3 prime strand. So let's write some base pair units also here, that is adenine, thymine, guanine, cytosine, adenine, thymine, guanine, cytosine, adenine. Okay, so what will be the complementary or next 5 prime 3 prime strand will be? It will be adenine pairs with thymine. Thymine pairs with adenine. Guanine pairs with cytosine. Cytosine pairs with guanine. Adenine pairs with thymine. Thymine pairs with adenine. Guanine pairs with cytosine. Cytosine pairs with guanine. Adenine pairs with thymine. Okay, so this is the DNA strand which is going to be undergoing what the transcription. Okay, so here, dear students, you have to keep in mind that 3 prime 5 prime position will be acting as the template strand. Okay, it will be acting as the template strand. Always keep in mind that 3 prime 5 prime strand will undergo the transcription process. That means the RNA will be synthesized from this particular strand. Okay, RNA will be synthesized from this particular strand. That is 3 prime 5 prime and 5 prime strand, and we will call that strand as template strand. Okay, next one, it is 5 prime 3 prime strand. Okay, 5 prime 3 prime strand. This strand is known as coding strand. What it is known as? It is known as coding strand. Okay, so this is the basic unit of a transcription. Transcription unit. The first one you have to note it down. It is the template strand and it is the coding strand. And also, dear students, it is very important to note down this. Important to note down the 3 prime 5 prime position and 5 prime 3 prime position. Okay, 3 prime 5 prime will be acting as the template strand, whereas in the case of 5 prime 3 prime will be acting as the coding strand. Let's see why it is known as coding strand on further next steps. Okay, so the next transcription unit includes, the basic transcription unit includes, okay, we can say that it is, first one you understood that it is DNA. Okay, second one we have to think about a promoter region. Okay, what it is known as? A promoter region. Promoter region means the transcription, the site of transcription, starting of transcription. It is the region where the polymerase enzyme will come and bind. Okay, so promoter region is the region where transcription starts. Okay, then third one, this is the basic structure. Okay, then the third unit, we can say, we cannot consider this as the unit. This is the important thing that should be there for transcription. So the first one we can consider as promoter. Okay, we can consider as promoter. Then next one we can consider as, so we can consider like this. Okay, next one we can consider as structural genes. Okay, inside DNA there is a region called structural gene where it undergoes transcription, complete transcription, and that particular site will give you the mRNA or the RNA. Okay, the third one is terminator region. Terminator site. Terminator site means where termination occurs. Okay, simple meaning. Initiation start. Promoter region is the region where the transcription start. Structural gene. The structural gene will give rise to the RNA. Okay, structural gene will give rise to RNA, whereas the terminator region, when the polymerase comes to the terminator side, what happens? The transcription stops. Okay, that we'll see what are the different steps in transcription. Okay, so this is the basic structural unit of transcription. Let's see. This is the basic structural unit of transcription unit. See, you can see. Okay, we can see a 3 prime 5 prime strand, and that 3 prime 5 prime strand is known as template strand. Okay, next one, it is 5 prime 3 prime strand, and that strand is known as coding strand. And here you can find a promoter side. Next, here you can find a terminator region. In between the promoter, see, this is the promoter side and this is the terminator side. In between, you can find a DNA strand, and that DNA strand is known as the structural gene. That particular strand, that flap in between the promoter region and the terminator region is known as structural gene. So this is the basic unit for transcription. From this structure, what is happening, what is providing, or what is produced? What is produced? An RNA is synthesized. Let's see how this RNA is synthesized. Okay, how this RNA is synthesized. Before going into that, we have to study some terms. That is gene. What is gene? It is the functional unit of inheritance. From the smaller class onwards, we are studying what is gene, right? What is gene? It is the functional unit of inheritance. Okay. In this, it is the DNA sequence coding for RNA molecule. Okay. In particularly, little more advanced, how we can define gene? We can define gene as the DNA sequence which codes for the RNA molecule. Okay, next, what is the cistron? It is very important. Okay, what is a cistron? Okay, what is a cistron? It is the segment of DNA which is coding for a polypeptide. Polypeptide means protein. Okay, polypeptide comes together to form a protein. So we can say those coding or those genes which codes for a polypeptide is known as cistron. Dear students, it is very important. So we can say we have two different kind of organisms, right? What are the two different kinds of organisms that you know? What are the two different kinds of organisms? Yes, you know about prokaryotes, right? Prokaryotes, right? Then you know about eukaryotes. Next, you know about eukaryotes. What is the difference between prokaryotes and eukaryotes? Prokaryotes and eukaryotes. See, in the case of prokaryotes, you have to understand there is no well-defined nucleus. Okay, there is no well-defined nucleus. Defined nucleus and cell organelles for prokaryotes. But in the case of eukaryotes, you have to understand that there is a well-defined nucleus. Okay, well-defined nucleus. There is well-defined nucleus and also there is well-defined cell organelles present in the case of eukaryotes. Okay, so if you are considering prokaryotes and eukaryotes, see, if you are considering a bacteria, okay, if you are considering a bacteria, the genetic material will be like this. So the genetic material will be like a chromatin. Okay, it is not completely present inside a particular cell organelle called nucleus. But if you are considering eukaryote, if you are considering a plant cell, okay, if you are considering a plant cell, inside a plant cell there will be a nucleus which is at the peripheral side. Due to the presence of a large vacuole, the nucleus will be present at the peripheral side. And inside the nucleus, what is present? The chromosome or the DNA will be present. Okay, so there is a specific structure called nucleus for the presence of the different genetic material. But in the case of prokaryotes, if you are considering prokaryotes, there is no particular cell organelle. Okay, there is no particular cell organelle. So we can say the the cystone which is present in the prokaryote and eukaryote is different. Okay, in the case of prokaryote and in the case of eukaryote, it is how, let's see how it will be. Okay, if you are looking into the structural gene, it is known as monocystronic structural genes in the case of eukaryotes. Okay, if you are considering eukaryote, what is the condition of cystone? The cystone condition is in monocystronic condition, which means it has exons and introns. Only one peptide will be formed at a time. Okay, and also it has split genes. It has split genes, which means it has exons and also it has introns. Okay, but whereas in the case of prokaryotes. Okay, whereas in the case of prokaryotes, these prokaryotes are polycystronic in nature. Okay, polycystronic in nature, which means among one cystone, different polypeptide, different genes for the synthesis of different polypeptides will be present. Okay, there is no exon, there is no intron. Okay, fine, understood, clear. If it is clear, you can give me a thumbs. Okay, so if you are considering prokaryotes are, dear students, it is very important, prokaryotes are polycystronic. Okay, they are polycystronic in nature, but whereas in the case of eukaryotes are monocystronic, and also they has the split gene with exons and introns. Okay, clear. And also one more thing you have to keep in remember, in the case of prokaryotes, I told you there is no specific organ to hold the genetic material. Okay, so we can say the transcription and translation. Okay, see, the transcription and translation occurs simultaneously. Okay, transcription and translation occurs simultaneously. Okay, whereas in this case, see, where does this genetic material is present? It is present inside the nucleus. Okay, genetic material is present inside the nucleus. So we can say the transcription, that means the formation of RNA, okay, the formation of RNA will be taking place inside the nucleus. Okay, whereas the process of formation of protein, that is translation will be taking place inside the cytoplasm. Okay, the site is very important. Okay, RNA is formed inside the nucleus. Okay, so when you have studied the structure of nucleus, you have learned about nuclear pore, right? Nuclear pore, you have studied the structure of nucleus in 9th grade. So, and also in 11th grade. So, when you studied the structure of nucleus, you have learned that there is nucleopore inside the nucleus. Okay, so after transcription, what happens? This mRNA or the RNA which is produced will be coming out of nucleus into the cytoplasm. Okay, so once it is reached into cytoplasm, what happens? Translation occurs and it forms into a protein. This you have to keep in mind. Okay, so let's move on the steps of transcription. What are the different steps of transcription? Okay, transcriptional unit we have already studied about monocystone and polycystone. Monocystonic structural genes are present in eukaryotes, whereas polycystonic structural genes are present in prokaryotes. And you know about difference between the prokaryote and the eukaryote. Okay, so if you are considering transcription, basically in prokaryotes, both prokaryotes and eukaryotes, there are 3 different steps. Okay, what are the 3 different steps? Let's see. It is initiation. Second step, it is elongation. Third step, it is termination. Okay, repeat. What is the first step? It is initiation. Okay, initiation means initiating the process. Okay, elongation means we'll come to know that nucleotides are adding. So this nucleotides, the process of adding nucleotide is known as elongation. So termination, I told you there is a terminator site. So when the polymerase enzyme which is binded over the DNA reaches into the terminator site, what happens? The process stops. Okay, so let's see in detail what is initiation. See, initiation, the enzyme known as RNA polymerase binds to the promoter site with sigma factor. Okay, so we can consider a DNA strand. We can consider a DNA strand. This is a DNA strand. Okay, so if you are considering this DNA strand, what happens? We have 3 dash 5, 3 prime, 5 prime direction, and also one is 5 prime, 3 prime direction. Okay, and also you know that there are two sides over there, that is a promoter side. Okay, and also you know there is a terminator side. Okay, in between you know there is a structural gene. Okay, this is the transcriptional unit. Okay, so what is initiation, dear students? What is initiation? It is the binding of DNA polymerase to the promoter site. Okay, so we can say this is the promoter site, right? This is the promoter site. So we can say it is the formation, or it is the binding of an enzyme. Okay, it is a binding of an enzyme to the promoter site. If a long chain is given, okay, if a two long chains is given and there is a promoter site, or our polymerase enzyme has come, they have searched all over. They have searched all over. Where is the promoter site? No, here it is. There is no promoter site. Then they will move to a next site. Is there any promoter site? They'll check. No, there is no promoter site. Again, they will start moving. Okay. So once they find the promoter site, they'll start the synthesis of RNA. So for example, we can consider here it as a promoter site. Our polymerase enzyme comes and it stands there. Then there is a sigma factor. It binds to the promoter region. Then it moves. Okay. It starts moving. When this enzyme starts moving, what happens? A new RNA will be synthesized. Polymerase enzyme means polymerase will add what it will be add? Nucleotides. Okay, you know what is nucleotide, right? So you understood. So this causes the local unwinding of the DNA of a double helix. Okay, one initiation happens, what happens? A small unwidening will be taking place, like in replication. Replication, you know that is there is a enzyme known as helicase which helps in the unwidening of the enzyme, unwidening of DNA. Okay, here what is the enzyme? Polymerase enzyme itself will be acting as itself will be acting as the unwidening enzyme. Okay, once it is unwidened, it will be moving forward. Then what happens? Nucleotide will be added. Okay, so there is an initiation factor, and that initiation factor is known as sigma factor. Okay, so sigma factor, they have searched everywhere and they came here with sigma factor. After unwidening, the sigma factor understood that there is a promoter region here. They will bind to that particular site and they'll start the synthesis. Okay, we can particularly say initiation means it is the attachment of the sigma factor towards the promoter region. Okay, clear? Also, it is the unwidening of DNA for transcription. Okay, clear? Clear, that 2 points is very important. You have to write it in your answer sheet when it is given for your exam. Okay, so next one, see, next step it is elongation. What is next step? It is elongation. What is elongation? You know, right? I told you that here if you are considering this is a promoter site or sigma factor comes and bind, then what happens? Then if it is moving towards the always it will be moving from 3 prime to 5 prime, so that the synthesis will be always in 5 prime 3 prime direction. Okay, first if you are considering this is the 3 prime 5 prime and this is 5 prime 3 prime. Okay, so we have already said that 3 prime 5 prime will be acting as the template strand. and 5 prime 3 prime will be acting as the coding strand. So what happens here, if this polymerase enzyme comes and bind, the synthesis will be taking place in 5 prime 3 prime direction. Synthesis will be taking place in 5 prime 3 prime direction. That means the first 5 prime will be coming out. Okay, 5 prime will be coming out, the 3 prime will be, when it reaches to the terminator side, the 3 prime will be stopped. 3 prime will also will come together, or it will also come along with the structure. I'll draw it once again in a plain sides. Okay, so if we are considering this as the two strands. Okay, so what is this strand known as? 3 prime 5 prime strand. This strand is known as 5 prime 3 prime strand. Okay, so what is the first step? First step is the initiation. Initiation step, what happens? Our polymerase enzyme comes and bind and they scan for the promoter site, and they understood that this is the site of promoter, and there is a sigma factor. The sigma factor will come and bind to the promoter region. Sigma factor means there is the C. You have to keep in remember, these are also a kind of RNAs. Okay, polymerase enzyme is also a kind of RNA. They have also sequences. Okay, they have also sequences. So we can say the sigma factor is also having the sequences. Since it is having the sequence, it can complement. Okay, it can complement to the 3 prime 5 prime direction. So this, it will be binding towards this 3 prime 5 prime region. What happens here? They will add the first nucleotide here. Okay, they'll start adding the first nucleotide here, the second nucleotide here, the third nucleotide here, fourth nucleotide here, fifth nucleotide here. Okay, and the bond will be formed. Then it will form sixth nucleotide here. Okay, it will be forming like when polymerase enzyme is moving. When polymerase enzyme is moving, the nucleotide is also formed. The nucleotide is also formed. So what is the second step? Elongation. Elongation means the polynucleotide will be elongated. Okay, 5 prime, 3 prime direction. So we can say this first nucleotide will be coming outside. Okay, second nucleotide, then third nucleotide. We can write the name so it will be easy for you to understand. Okay, adenine, guanine, cytosine, uracil, adenine, guanine, cytosine, uracil. Okay, so this coming, the RNA which is coming out will have 5 prime end, and this is having the 3 prime end. Okay, the RNA coming out will have 5 prime end, and the DNA RNA it is having which is inside, it will have 3 prime end. So when the polymerase enzyme, so we can consider this is the terminator site. When it meets the terminator site, polymerase enzyme, another protein molecule known as Rho. Okay, Rho factor. What is known as? This is known as Rho factor. Rho factor will come and bind to this structure. Okay, that particular terminator site. When Rho factor comes to the terminator site, that code a signal, the transcription has to stop. Okay, the transcription has to stop. So what happens? The transcription will be stopping. Later on, the sequence will be coming out as 5 prime. The sequence will be coming out as 5 prime, 3 prime direction. Dear students, again I'll write another in a plain slide. Okay, so dear students, we have seen that this is 3 prime end and this is 5 prime end. So we can write AUG, not AUG. Okay, just we can write adenine, thymine, guanine, cytosine, adenine, thymine, guanine, cytosine, adenine, thymine. Okay, so next one, it is 5 prime 3 prime strand. Okay, so this strand is having thymine, adenine, cytosine, guanine, thymine, adenine, cytosine, guanine, thymine, and adenine. Okay, this is the strand. So let's consider from thymine and adenine what is the promoter site. This is the promoter site. Let's consider this is the promoter site and this is the we can say CA will be the terminator site. Okay, so what happens? This strand which is present in between. Okay, the strand which is present in between this 3 strand, 3 prime and 5 prime. See, it is G, C, A, T, G, C. Okay, this strand has to be transcribed. This strand has to be transcribed. So, you know that 3 prime, 5 prime will be acting as 3 prime, 5 prime will be acting as the template strand. It will be acting as the template strand, and 5 prime, 3 prime will be acting as coding strand. Let's study why it is known as coding strand. Okay, so what happens here? As we told, the polymerase enzyme will be coming and it will bind over the TA region. So we can say polymerase enzyme comes and binds here. Okay, polymerase enzyme, especially the sigma factor, comes and binds to this particular region. So what happens? The synthesis will be starting. Okay, that means the nucleotide has to be added. Right? What is the next nucleotide present here? It is guanine. Okay, so in guanine, who will be complementing guanine? It is cytosine. Okay, who will be complementing guanine? It is cytosine. So we can write it as cytosine. Okay, who will be complementing? Cytosine will be complementing. Then here cytosine, who will be complementing? So this polymerase enzyme comes here. It gives cytosine here. Then it moves towards next side and cytosine is here. Who will be complementing? Guanine will be complementing. Polymerase enzyme will give guanine. It will put guanine. So my dear students, next one it is adenine. Adenine will be giving instead of adenine, or adenine will be pairing to thymine. Thymine, not thymine. In RNA, there is no thymine. Instead of thymine, there is uracil. Okay, so thymine, again, here comes thymine. So we can say adenine. Guanine, here, what is guanine? It is cytosine. Okay, now this is the mRNA which is synthesized. Okay, this is the mRNA which is synthesized, and you know that it is 3, so not 3 prime. It is synthesized in which prime? It is 5 prime, and this is 3 prime. So we can say 5 prime will be coming out. Okay, this is the basic concept, so you have to be thorough. Okay, so 5 prime, C will be first coming out, G will be next coming, then U, then A, then C. Okay, so we can say this is 5 prime and this is 3 prime. When it reaches to this CA, I told you that this will be considered as the terminator site. Okay, this will be considered as the terminator site. So when it reaches to the terminator site, who will be coming? A raw protein will be coming. Okay, sorry, this is sigma, this is raw. Okay, raw factor will be coming, and this raw factor will come and attach to the terminator site. When this raw factor comes and attaches, what happens? The polymerase enzyme stops the synthesis. So you got an mRNA here, and what is that mRNA? What is the codons, or what are the different nitrogen base which is present in this mRNA? CGUAC. Okay, it is CGUAC. Dear students, now look at the coding strand. Now look at the coding strand. If you are considering coding strand, it is TAC. We have started the structural genes from here. Okay, so it is C. Okay, it is C. The newly synthesized mRNA is also having C. Okay, the newly synthesized mRNA is having C. Next one, it is G. Okay, so next one, it is G. The newly synthesized RNA is having G. What is the strand here? The coding strand is having G. Okay, uracil in the case of RNA, but if you are considering the DNA, what is present? P is present. A, this is A, and instead of the coding strand, you can find A. So next you can find C. In the coding strand also, you can find the C. That means the newly synthesized mRNA, the newly synthesized RNA, and the strand which is known as 5 prime 3 prime strand. Okay, the newly synthesized RNA and the 5 prime 3 prime strand. Okay, strand in DNA looks similar except in thymine and uracil. Okay, except in thymine and uracil, you can find the same strand, the exact same strand. That is why we call the 5 prime 3 prime as coding strand. Okay, that is why we call the 5 prime 3 prime as the coding strand. Okay, you got it? You got it? So that is all about the transcription, steps involved in transcription. What is the first step involved in transcription? What is the first step involved in transcription? It is initiation. Okay, it is initiation. We can say in initiation, what is the sigma factor? Okay, sigma factor is present in polymerase, and this polymerase will come and attaches to polymerase is there. Polymerase enzyme is there. This polymerase enzyme will come and attaches to the which region? It comes and attaches to the promoter site. Okay, clear? Then next one it is elongation. Next step it is elongation. So what happens in elongation? In elongation, addition of nucleotides. Okay, addition of different nucleotides. Okay, different nucleotides. Nucleotides including adenine, guanine, uracil and cytosine. And also you have to keep in remember that they will be inactivated form, which means they are in a phosphorylated condition. Okay, next one it is termination. Next one it is termination. What happens in termination? What happens in termination? In termination, the transcription stops. Okay, transcription stops. Who comes here? Who comes here? Rho factor. Rho factor comes here and stop the transcription. Okay. So this is the conclusion for transcription. There are 3 important steps and what happens in 3 steps. You have to keep on reading. Okay. Read the textbook well. These are the basic things. Okay. So next you have to understand the difference between prokaryotic transcription and eukaryotic transcription. Okay. The basic things happening in prokaryote and eukaryotic will be the same. That means it is initiation, it is elongation and it is termination. Okay. But we have studied in the case of what we can say, in the case of eukaryotes are monocystronic with split genes, right? Split genes means exons and introns. So this exons are coding sequences and introns have been considered as non-coding sequence. Okay, exons and introns is present, and this exons and introns have to be removed. Okay, so we can say, let's see the steps involved in eukaryotes. Before moving into eukaryotes, you just look into the initiation, elongation and termination process. See, this is the sigma factor. Sigma factor comes and joins to the promoter region. Elongation, along with this sigma factor will be removed. Thus, polymerase enzyme will be moving on. Here what happens? An enzyme. This mRNA will be synthesized and the newly synthesized mRNA is in which direction? 5 prime, 3 prime position. Okay. This is a newly synthesized mRNA after termination. That is 5 prime, 3 prime position. Okay. Always keep in mind the synthesis will be taking place in 5 prime, 3 prime direction. Okay. Next we have to study about eukaryotic transcription. The same process will be taking place in eukaryotic transcription, but you can find the 2 different structures, that is introns and exons. Okay, so what is present? Since eukaryotes are monocystronic, since eukaryotes are, we have studied eukaryotes. Okay, eukaryotes. In eukaryote, what is the speciality? What is the speciality? We have told that they are monocystronic. Okay, they are monocystronic. Monocystronic in sense they have split genes. They have split genes. Okay, and what they possess? They possess exons and introns. Okay, they have exons and introns. Okay, so if we are considering exons are needed, or we can say they are the coding sequences. Okay, they are the coding sequences. Okay, whereas in the case of introns are the non-coding sequences. It is not needed for translation. Okay, non-coding sequences. Okay, the same process happens in eukaryotes also. Initiation process will be taking place, then elongation will be taking place, then termination will also takes place. Okay, so after this, what is the heterozygous, heterogeneous RNA will be synthesized, and that RNA will contain this exons and introns. Since it is a mix of exons and introns, we just need for translation. For translation, we just needs exons. Okay, we just needs exons. And also you have to keep in remember that, dear students, the transcription, okay, the transcription takes place in nucleus. Okay, nucleus. After transcription, what is obtained? RNA is obtained. Later on, this RNA is transported into cytoplasm. Why it is transported to cytoplasm? It is transported to cytoplasm for protein synthesis. Protein synthesis. Everything you have to correlate, okay? Protein synthesis. Okay, for protein synthesis, we need only exons. We need only exons. We don't want introns. But when it is formed, when this RNA is formed, it is a mix of introns and exons. Okay, so here what we have to do, we have to remove the introns. We have to remove this introns and we have to transport this exons to cytoplasm. That is a task. Okay, that is a task. So what happens here? This transcription in eukaryotes will be taking place in the similar method. That means initiation, elongation and termination will be taking place. Along with that, the three important steps also will be taking place. Okay, what are those three steps? What are those three steps? The three steps involves, first one, splicing. Okay, splicing. Okay, second one, it is capping. Okay, giving cap. You will see capping. Okay, third one, it is tailing. Giving some tails. Let's see what is this capping and what is this tailing. And also splicing. Okay, dear students, you know that when RNA is formed, it is with exons. It is with exons and introns, right? And I told you exons is only needed, introns are not needed. Okay, so we can say the removal of introns. Okay, removal of introns from the whole sequence. Okay, removal of introns from the whole sequence which is having exons and introns is known as splicing. Simple. Okay, so if you are considering a DNA, if you are considering an RNA, see this is RNA. This is an RNA. Okay. Okay, this is an RNA. This is an RNA which is synthesized after transcription. So this is exon 1, this is exon 2, and this is exon 3. Okay, there are 3 exons here and 2 introns. Okay, this is intron 1 and this is intron 2. Here you have 2 introns and you have 3 exons. So the removal of the 2 introns, okay, the removal of 2 introns to get a complete RNA, with 3 exons, This is exon 1, this is exon 2 and this is exon 3. Here there is no introns. Okay, there is no introns. That process is known as splicing. Okay, so now it is clear. What is splicing? It is the removal of introns. It is the removal of introns from hnRNA. Removal of introns. Okay. So next one it is capping. The second process is capping, right? So you know the transcription takes place inside. Transcription takes place inside. Transcription takes place inside. Yes, transcription takes place inside the cytoplasm. Sorry, in nucleus. Okay, it has to move towards which region? Cytoplasm. But when it is moving towards the cytoplasm, there will be some kind of attack. Okay, so in order to compensate that, there is one particular enzyme, there is one particular molecule which is attaching, which is acting as cap towards this mRNA. Okay, and that molecule is known as C. It is methyl guanosine triphosphate. Okay, what is that? Methyl guanosine triphosphate. It is methyl guanosine triphosphate. So it gets a cap. So if you are considering an RNA, see, this is an RNA which is coming out. Okay, the transcription is still going on. Okay, transcription is still going on. So this is the 5' end which has come out of the DNA. Okay, if we are considering DNA, we can draw the DNA too, then it will be clear for you. Okay, if this is DNA, this is DNA. If this is DNA, what happens? It comes. Okay, 5', 3' end. The RNA will be coming out. So, in order to protect from the exonuclease and endonuclease reactions, what is giving? We are giving a molecule known as C. On 5', what is present? It is present methyl guanosine triphosphate. Methyl guanosine triphosphate. It will be acting as a cap. Okay. Next we have the 3' end. Okay. Next you have 3' end, right? So in this 3' end, what will be acting as? Polyadenylation will be given. That means a long chain of adenine will be giving. Okay. See, this one is long chain of adenine molecules. So that is tailing. That process is known as tailing. Okay, so in the case of transcription in eukaryotes, there are three extra process taking place. What are the 3 extra process taking place? First one, it is splicing. Okay, you have learned about splicing, understood. I think you are clear with splicing. What is splicing? Splicing, it is the process of removal of introns. Okay, removal of introns to make exon complete and exon complete RNA. Then second step it is capping. It is the addition of methyl guanosine triphosphate. Then the third step is tailing. What is tailing? It is adding many number of adenine molecule towards the 3' end. So now it is protected. Okay, now it has a cap, it has a tail, it is protected, so it can safely move into the cytoplasm. Okay, once it reaches to the cytoplasm, what happens? Once it reaches to the cytoplasm, translation will be occurring. So that is all about transcription in prokaryotes and transcription in eukaryotes. Okay, once again, if you are looking into the transcription, you have to think about the structural unit. Okay, what is the unit for transcription? It includes DNA, and among DNA, the promoter site is there, structural gene site is there, then terminator site is there. Then coming into the process, it is initiation, then elongation, then third step is termination. Okay, then moving into the difference between the prokaryotic and eukaryotic transcription, eukaryotes have 3 more steps, that is splicing. Then second one it is capping. The third one it is tailing. Okay, that's all about transcription. Okay, next we have to learn about Translation. Okay, what is that? Translation. For translation, we have to understand what are the requirements. Okay, what are the requirements for translation? Okay, so for that you have to study about the mRNA. For translation, what are the requirements? The requirements include mRNA. Okay, mRNA is required. Then what is required? Amino acids are required. Okay, amino acids are required. These amino acids are present inside the cytoplasm. Okay, as amino acid pool. Okay, then we have to get ribosomes. Okay, you have learned the structure of ribosome previous year, right? In grade 11. Prokaryotic ribosome and eukaryotic ribosome. Ribosome have two different subunit. That is a larger subunit and a smaller subunit, right? Then you need a tRNA. Okay, what is tRNA? tRNA means transfer RNA. Okay, what is tRNA? Transfer RNA. Okay, transfer RNA. So these are the essential components for the process of translation. What it is? mRNA is needed. Then mRNA, it will be coding to something and that will form amino acids. Amino acid pool is there inside the cytoplasm. So we have to understand about amino acid. Amino acid is required. Okay, who is taking amino acid from the amino acid pool towards the mRNA, tRNA? So we will call the tRNA as transfer RNA. The next thing for the process of translation, what is important? Ribosome is important. Ribosome will be acting as the what it is? Major component, and it has a larger and smaller subunit. Let's see each and everything. mRNA, you understand it is messenger RNA. mRNA means messenger RNA, and this mRNA is having a codon or sequence. Okay, so next we will study about the genetic codon and the amino acid. Okay, so what is genetic code? What is genetic code? See, there is a mRNA. mRNA means messenger RNA. It gives the information, right? So we can say genetic code, it is a sequence of nucleotide in mRNA which contains information for protein synthesis. Messenger RNA, message. Okay, it gives the message. What kind of message it is giving? It is giving an information for the protein synthesis. Okay, what it is giving? It is giving the information for protein synthesis, and that process is known as translation. Okay, and that process is known as translation. Okay, so we have to study the salient features of genetic code. Okay, always keep in mind that genetic code is a triplet code. What it is? It is a triplet code. If you are considering an mRNA, see, we can say adenine, uracil, guanine, cytosine, uracil, adenine, guanine, cytosine, uracil, adenine, guanine. Okay, so the first three will be considered as first triplet. The next three will be considered as the next triplet. Then later on, this three will be considered as one triplet, and we have one more triplet. Okay, so these are the triplets. Okay, so what is the speciality of genetic code? Genetic code is a triplet code. Triplet code. What is the first triplet code which is given here? It is AUG. What is the second triplet which is given here? It is CUA. What is the next triplet code given here? It is GCU. What is the next triplet codon which is given here? It is AGU. Okay, each genetic code is written as triplet code. Okay, so we can say the three codons do not code for any amino acids. Okay, so there are three different codons, and these codons will be acting as the stop codons. What codons it will be acting as? It will be acting as stop codons for termination or termination in translation. Okay, what are the three codons? That it is it is UAA. Okay, UAA. Then next one UAG. Okay, next one UAG and one more is there UGA. We will call it as UGA. Okay, what we will call? We will call it as UGA. Okay, UGA and one more is there, that is UAG. Okay, so please note it down. It is important for exam. It is clearly they are asking what are the three different stop codons. So you have to mention that it is UA, UGA and UAG. Okay, so next you have to stand. They function as stop codon. Okay, these functions as stop codons or termination codons. Okay, so next point, genetic code is universal. Okay, if you are considering genetic codon is similar for every organism. Every organism, from a simple prokaryotic organism to a complex eukaryotic organism, it is similar for every organisms. Okay, so some exceptions are found in mitochondrial codons in some protozoans. There are some exceptions only in the case of mitochondrial codons. What is mitochondria? You know the structure of mitochondria, right? The mitochondria, if you're looking into mitochondria, they have its own DNA. So when we are considering the codons in mitochondria, some exceptions are there for the mitochondrial codons in some protons. There are some differences. Okay, then no punctuations between adjacent codon. That's why we'll call it as comaless. Comaless code. There is no first three triplet is there. There is no comma in between. The next codon will be moving on. Okay, then the codon is read in mRNA in a continuous fashion. It is moving in a continuous fashion. That means there is no stoppage in between. There is no hyphen. There is no slash. There is no comma. Nothing is there. Okay, it is reading in a continuous fashion. Okay, genetic code is non-overlapping. Never it overlaps. Okay, never it overlaps. So we can say genetic codon is non-overlapping. Okay, a single amino acid is represented by many codons. For example, if you are considering leucine. Okay, if you are considering leucine, this leucine will have many codons. I'll show the image so it will be more clear for you. Okay, so here you can find leucine, right? See, CUU, I'll write here for leucine. CUU is one codon, another one is CUC, the next one is CUA, the next one is CUG. Okay, every four codon, this four codons is coding for one particular amino acid, that is leucine. Okay, that is leucine. Then if you are considering proline, you can say that it is CCU is coding for proline, CCC is coding for proline, CCA is coding for proline, CCG is coding for proline. This everything is coding for proline. Okay, so that means different codons is present. Different codons are coding for one particular amino acid. Okay, it is coding for one particular amino acid. Okay, such codons are called degenerate codons. We'll call that codons as degenerate codons. And there is an exception for methionine and tryptophan. Dear students, CBSE have a tendency to ask the exceptions in particular. So you have to study what is coding for AUG and what is coding for UGG. Okay, AUG and UGG. AUG codes for methionine. Methionine. Okay, it is the initiation codon. It is the starting codon. It is a starting codon both in the case of prokaryote and eukaryote. Whereas UGG, if you are considering UGG, this UGG is coding for one aromatic amino acid, that is tryptophan. Okay, it is coding for tryptophan. Okay, it is very important. Then genetic code is unambiguous and specific. Always genetic code is specific. Okay, it won't move for anything. If AUG is coding, it is coding for methionine. If UGG is coding, it is coding for tryptophan. UGG won't code for methionine. UGG won't code for any other amino acid. If UGG is present, UGG will code only for tryptophan. Okay, then that is one codon specifies for only one amino acid. Okay, if you are considering, so you will have some doubt in the case of previous statement that one particular amino acid have different codons. Okay, but there is one thing, that particular sequence. Okay, that particular sequence will code only for one particular amino acid. Okay, that codon will code only for one particular amino acid. See, if you are considering CCU will be coding only for proline. Okay, it won't code for tryptophan. It won't code for methionine. Okay, you understand? Is it clear? Okay, so CCU codes for proline. CCC will be always coding for proline. CCA also will be coding for proline. CCG also codes for proline. Okay, that is the condition here. That is the condition here. Okay. So we can say the one codon specifies for only one particular amino acid. Okay. What is the next point? The codon reads in 5 prime 3 prime direction. Again, the direction is specified. It codes from 5 prime 3 prime direction. Dear students, polymerization occurs only with 5 prime 3 prime direction, and one more thing, the codon reads only in 5 prime 3 prime direction. These directions are very important. Okay, directions are very important. Keep in mind that 3 prime 5 prime of a DNA strand in transcription codes for template, and the other one will codes for the coding strand. Okay, these are simple things that which you may lose your marks. Okay, then next one, it is an important thing. Put it in a box and write in your notebook. Okay, AUG, as I have mentioned before, AUG acts as an initiator codon. Okay, AUG acts as an initiator codon. AUG acts as an initiator codon. What is initiator codon? In the process, in the coming class, we will come to know it has an initiation process. Okay, it has an elongation process, it has a termination process. So we can say it is the initiator codon. If mRNA, if it is a long mRNA, then we can say what this if AUG is present in the mRNA, it will be acting as the initiator codon. If it codes that particular sequence, AUG is present, it will be coding only for one sequence, and that sequence is methionine. Okay, methionine. It will be added with formyl methionine. We will call FMET. FMET will be added for every translation initiation step. Okay, so is it clear? It is very important. AUG, it has dual function. It acts as an initiator codon. Also, it acts as it codes for methionine. Okay, methionine is an amino acid. Okay, so next we have to study, this is all about the genetic codon. You have to study the specific characteristic features of the genetic codon, and let's see how this genetic codon helps in the translation, and also what is the structure of tRNA. Okay, as I have told, what are the requirements? RNA is a requirement, mRNA is required. Then next we have to get amino acid. Now you understand how amino acid will be formed in amino acid pool and how it will be coding for one sequence. Okay, the next important thing is tRNA. Okay, transfer RNA. See the structure of tRNA, it is like a clover leaf. The 2D structure of a tRNA will be in a clover leaf condition. Okay. What is this clover leaf condition? See, the tRNA molecule has about 60% of its part double stranded, and the rest remains single stranded, which has unpaired base. Okay, so we can say this is the structure of tRNA. It has different sites. Okay, there is anticodon site. Okay, this tRNA is having an anticodon site and also an amino acid attachment site. Then we have 5 prime 3 prime site. Okay, let's see what are the different sites for tRNA. Okay, so there is an anticodon site. Okay, there is an anticodon loop. For tRNA, what is there? There is an anticodon loop that has bases complementary to codon, which it attaches to the mRNA. Okay, so if there is a codon, see, this is the anticodon site. This is the anticodon site. Okay, where AGU is the codon in mRNA. If AGU is the codon, then anticodon should be UCA. Okay, anticodon should be UCA. So what is the next structure? Then amino acid acceptor end will be there, and this amino acid acceptor end is in 3'N. So if you are drawing the structure, okay, this is 5' 3' end, and it has an anticodon site. Okay, so we can say this is the anticodon region. Okay, this is the anticodon region. And amino acid end is there, and it is attached where it is attached? It is attached to the 3'N. Okay, so we can say this is an amino acid end. Here we can draw a structure of, okay, this is an amino acid end where it carries the amino acid. Okay, next one, see the 5' end is there. This 5' end bears the guanine group. Okay, the guanine group, it is having the guanine group on its 5' end. Okay, then we have two more loops, that is T-size loop and the HU loop. Okay, we can consider this as T-size loop. T-size loop, and this is the HU loop. So these are the two different loops. Okay, these are the two different loops that you can find over the structure of a tRNA. Okay, so if you are looking into the structure of tRNA has, you have to keep in remember that there is an anticodon site. That is a very important anticodon site. Then you have to keep in remember on the 3' position, what is there? Amino acid is there. Okay, that is the region where amino acid is attached to the transfer RNA from amino acid pool. Okay, then on the 5' end, there is a guanine group, and there is two regions, two overlapping region, and that overlapping region is known as T-size region and DHU region, where it attaches with ribosome. Okay, in T-size loop, ribosome is attached. This has some unusual base pairs. Okay, and what are the unusual base pair present? It is pseudouridine. It is very important. It is given in bracket, but it is very important. Okay, and ribothymidine. Okay, then DHU loop, it is the binding site for the amino acyl synthetase. Okay, it is the site for amino acyl synthetase, attachment for amino acyl synthetase for the activation of this particular tRNA molecule. Okay, so that is the structure of tRNA. We have find the structure of mRNA or just the sequences of mRNA. Then we understood the amino acid. Okay, then we understood about the structure of tRNA. The next process is translation, and we will study translation in the next session. Okay, we'll study translation next session. Translation, and along with translation, we will study the operons too, Human Genome Project and DNA fingerprinting in the next coming session. So be thorough with this much topic. Okay, okay. So on the coming class, Juhima will be coming with the live. Juhima will be coming with the live for, okay, on the next Monday, that means on coming 11th of this month, Juhima will be coming with the same chapter which was we were previously taking. Okay, and you have to be on time for the live session. Thank you so much. We'll see in the next class with translation, the complete process of translation, and also with lack of problem. Thank you.